Electron microscopy is part of cellular pathology, and unlike routine histology, which looks at the overall architecture of tissue sections using a light microscope, EM can examine ultrastructural features within the cell using a transmission electron microscope. The EM team processed and reported approximately 1,800 samples last year. Many different types of EM samples are received into the laboratory. They are checked, booked in, and ready to be processed for examination. The samples are processed using an EM processor, impregnating the tissue with resin, which will allow very thin sections of the tissue to be examined by transmission electron microscopy. This is different to the wax processing in routine histology. The tissue is embedded in liquid resin in shaped moulds, and the resin is allowed to harden overnight. The result is a block of hard resin with a small portion of tissue ready for sectioning. The blocks are sectioned using an ultramicrotome to produce very thin sections containing tissue. These sections are stained and ready for examination. This is our transmission electron microscope. The microscope uses electrons to create an electron beam which passes through the stained tissue sections. This creates an image of the tissue which is captured using a digital camera. Electron microscopy produces ultrastructural reports which are one piece of a diagnostic puzzle. As an ancillary test, it can provide supportive information, and in some cases it can even provide diagnostic information that no other test can. EM can also be used to identify other features like bacteria and viruses. Our EM team participated in research work surrounding the identification of the coronavirus in cilia samples. This involved processing and examining infected and control samples to identify what the virus looked like and what effects the virus had on the cell. These images shown are taken from the study, and the structures seen are consistent with images of virus reported in journals around the world. Electron microscopy is a highly specialised technique requiring specialist equipment and trained biomedical scientists and biomedical assistants. We look at a variety of tissue types and are one of the largest diagnostic centres out of less than 30 in the UK. Thanks for listening.